everyone, thanks for having us here. And let me do a simple introduction. Here standing next to me is Mr. Bilu Huang, and he is our chief scientist of Fuzhuang Therapeutics, and I am the COO, and I will be speaking for Bilu Huang, and when it comes to the Q&A section, I will be interpreting for Bilu Huang. And first, thanks for having us on this prestigious summit. So today, we are going to introduce why do we age. This is the unified theory of aging proposed by Bilu Huang in 2021. The telomere DNA and ribosomal DNA co-regulation model for cell senescence. It can also be called TRCS model. First, as the declaration of interest statement, Bilu Huang is the founder and the CSO of Fuzhuang Therapeutics, and me, Wen, or you can also call me Claire. I am an employee at Fuzhuang Therapeutics. Einstein once said, if you cannot explain it simply, you do not understand it well enough. And as we all know, aging and death are so frightening. So why do we age? Aging is programmatically controlled as a program, not the result of random damage accumulation. The theories of aging are divided into two categories, the damage accumulation theory and the programmed aging theory. When 3 killifish strains, one with a 3-month lifespan, one with a 9-month lifespan, and one with a 16-month lifespan, they evolved under raining seasons of corresponding length, and they are reared together in the same aquarium. Their lifespan difference remained. This shows that post-puberty aging is still genetically programmed, because random damage accumulation cannot explain why these 3 closely related, almost identical species differ so dramatically in longevity, nor why these differences match the duration of the raining seasons in their native habitats. Also, since antioxidants can reduce various types of damage, but have failed to extend the lifespan of wild-type mice, this evidence alone is already sufficient enough to refute the theory that aging is caused by the accumulation of various random damage. 2. Individual aging is caused by the replicative senescence of adult stem cells. Replicative senescence is characterized by the phenomenon that with each cell division, daughter cells become older than the mother cell. Specifically, this involves downregulation of overall protein and ATP synthesis rates, while the expression of some proteins is upregulated and others downregulated. This leads to a decline and alteration in cellular function, indicating that cellular senescence is also programmed, continuing until cell cease division upon reaching the Hayflick limit. The somatic cells constituting the various tissues and organs of an individual are broadly classified into 2 categories, adult stem cells and functional cells. Although no adult stem cells have been identified in the heart, cardiomyocytes can renew themselves through self-replication. Therefore, cardiomyocytes serve as both adult stem cells and functional cells. Both adult stem cells and functional cells can be cleared by immune system due to factors such as cellular senescence, genetic mutations, or viral infections. Then they are replenished through the cell replication and differentiation of adult stem cells. However, the adult stem cells themselves undergo replicative senescence due to the repeated self-replication. Similarly, the functional cells differentiated from aged adult stem cells are also aged functional cells, leading to the gradual aging of tissues, organs, systems, and ultimately the entire individual. Therefore, the fundamental cause of us as the individual aging can be ultimately attributed to the replicative senescence of adult stem cells. And we can take a look at this figure 1. Individual aging results from replicative senescence of adult stem cells. With each replication of somatic stem cells, the daughter cells age slightly compared to the previous generation. Consequently, the functional cells differentiated from these aging somatic stem cells are also aged, like they age after each passage and self-division, leading to the progressive aging of tissues, organs, systems, and ultimately the entire individual. 3. The replicative senescence of cells is co-regulated by telomere and rDNA. Telomeres are tandemly repeated multi-copy DNA arrays, and telomere shortening has been proven to act as a countdown timer for cell replication and senescence. However, in some cells where telomeres do not shorten, the number of cell replications and lifespan remain limited. Furthermore, it has been observed that maintaining telomere length with telomeres, which increases the number of replications, ultimately does not prevent cells from ceasing replication and dying. Therefore, besides telomeres, there must be another multi-copy tandemly repeated DNA arrays in the nucleus that serves as a second countdown timer for cell replication and lifespan. Consequently, Bilu proposed the telomere DNA and ribosomal DNA co-regulation model for cell senescence, TRCS. This theory posits that the shortening of telomere and/or rDNA arrays leads to the increased level in the tumor suppressor protein, p53, thereby inducing cellular senescence. Furthermore, the TRCS theory has been experimentally validated as correct, with rDNA contributing a greater weight to senescence than telomeres. And here is the TRCS model. In the left, the chromosomes with long arrays of telomeres and rDNA, and p53 is rapidly degraded. p53 levels are low, and the cell is young, useful. In the right, chromosomes with short arrays of telomere and rDNA, and p53 is slowly degraded. p53 levels are high, and the cell is aged. And the evidence for the TRCS aging model. The validity of the theory depends on whether a theory is self-consistent, since the shortening of telomere DNA and rDNA arrays are the fundamental cause of cell aging and the countdown substance that drives the programmed expression of the gene groups. Then the telomere DNA and rDNA that is consumed in somatic cells must be replenished in early embryonic cells or germ cells. Otherwise, life cannot be perpetuated through generations. Fortunately, there are already enough evidence that have shown that telomere and rDNA consumed in somatic cells can be replenished in early embryonic cells and germ cells. Stem cells in mice can experience rDNA array shortening due to the mTOR1 activation. Rapamycin, an anti-aging drug, can inhibit mTOR1, and thereby suppressing rDNA transcription and cell replication, slowing down replicative cellular senescence, and extending the lifespan of mice. It is almost the only most effective anti-aging method or drug that has been proved can extend the lifespan of mice by the most rigorous and authoritative ITP test. So therefore, from the perspective of first principles, the lifespan of any species is determined by the shortening rate of telomere and rDNA arrays. The TRCS model has previously pointed out that the shortening of telomere and or rDNA arrays leads to an increase in the level of tumor suppressor p53, protein p53, causing cells to enter a state of senescence, and the mechanisms of rejuvenation through the pluripotent reprogramming is due to the significant extension of telomere and rDNA arrays. Accordingly, we knocked down the 45S rDNA copy number in primary mouse and human cells, and the results showed that the aging biomarkers p53, p21, p16, and SA beta gal were significantly upregulated as expected by Bilu Huang, while telomere length, cell viability, and cell passage numbers were significantly reduced. We also tested aging cells and human ES cells and human iPS cells. And we found that the telomere length and 45S rDNA copy numbers in aging cells were significantly reduced, while those in human ES cells and human iPS cells, that's as known as the young cells, they were significantly increased. So this data strongly demonstrates that the rejuvenation mechanism of ES and iPS is not due to the epigenetic reprogramming, but because the length of the telomere DNA array and the 45S rDNA array has significantly increased or reset. The fundamental cause of cellular aging and high flick limit is co-regulated by telomeres and 45S rDNA. And the weight of rDNA in aging is greater than that of telomeres. So it's already validated to be correct by experiments, this aging model. 5. A unified picture explaining the 11 hallmarks of aging. Since the telomeres and the rDNA are universal among all organisms, from yeast to nematode to mice to humans, therefore the mechanisms of aging across various species have been unified. Moreover, the 11 other acknowledged, well-established hallmarks of aging that has been shown in the very famous paper in Cell journal, hallmarks of aging, the expanding universe, that have been concluded. They are also the downstream consequences mediated by telomere and rDNA shortening through the p53 pathway, such as genomic instability, chronic inflammation, epigenetic alteration, and mitochondrial dysfunction. So they are all the downstreams of the upregulation of p53 protein that's caused by the shortening of telomere and rDNA. The table shows that the 11 hallmarks of aging mediated by telomere and rDNA arrays shortening through p53. And here's the recommended reading that Bilu published the causality of aging hallmarks in Aging and Disease this year. In summary, the ultimate cause of individual aging is the progressive shortening of telomere and rDNA arrays in tissue-resistant adult stem cells. In theory, replacing the aged adult stem cells in aging tissues with young adult stem cells can rejuvenate the aged tissues. However, transplanted allogenic stem cells will be cleared by the immune system within a week. Transplanting autologous stem cells that have been expanded in vitro is also useless, as the expansion process induces replicative senescence. Additionally, transplanting adult stem cells that are derived from iPS cells generated from our cells is also useless, because these cells often harbor DNA damage exceeding 70%, which also leads to their immune clearance. Therefore, these approaches are all not feasible. The only viable solution is to increase the length of telomeres and rDNA arrays in our adult stem cells. Besides this, apart from this, interventions at the metabolic level and in signaling pathways can only slightly extend lifespan, come with significant side effects, and are impossible to reverse aging. That is just like the ripamycin, which is the only most effective small molecular anti-aging drugs that have been tested by the ITP program. And if you want to learn and discuss more about aging, feel free to contact Bilu Huang. You can email to Bilu or connect with Bilu on LinkedIn or follow him on the X Twitter. And that's all. Thanks for the listening. And once again, thanks for having us on this very famous and very informative summit.
Anyone have any question related to the presentation?
I am a little bit confused on the adult stem cells and the stem cells that that mentioned that was 70% damage and wouldn't work. What are the differences?
Oh, so when I say if that's the stem cell, that's other people's stem cell, and you have it transplanted into your body, and it will be cleared by the immune system within a week. And when I said that the 70% damage, that is according to many papers that says the adult stem cells derived from iPS cells from our own body also have DNA mutations that it has also exceeded 70%. So it will also ultimately be cleared by the immune system. And that is exactly why there have already been almost 20 years since the first discovery of iPS cells, and it has not been used to replace our aged cells and to extend our lifespan. Just it's the very, very important thing is it will be cleared by the immune system. And also the Sumitomo Pharmaceuticals has recently discovered R&D iPS cell therapy that has been approved by the Japan FDA to treat the Parkinson's disease. But we have to be very careful. It also has been used with the immune suppressor, so like ripamycin. So it also indicates that it will be if we do not use the immune suppressants, it will ultimately be cleared by the immune system, those iPS cell therapy. So, is it clear?
But then what specifically is the adult stem cells that you refer to that would be ideal, that would work?
So we just mentioned the only viable way is to increase the telomere and rDNA in our own stem cells. That way they have no immune clearance problem and won't be cleared by the immune system, because the 99% of the biologics therapy medication will fail just simply because they will be cleared by the immune system, like the gene therapy, iPS cell therapy, like that.
Okay. So then, is there any strategies to improve the telomeres and the rDNA race?
Yes, for the telomeres, to extend telomeres, there are telomerase therapy, and not the small molecular telomere activators that won't work, but the telomerase therapy, the large molecular, as there are already several pipelines of that is being developed around the biotechs, not only by us but also around the globe. For example, our friend Professor Michael Fossil's biotech company Telosite, his company is now developing telomerase therapy to treating the Alzheimer's disease and cardiovascular disease. And rejuvenative technologies, they are using the telomerase therapy to treating some age-related degenerative disease in the lung and the liver. As for the rDNA, just how to increase rDNA in our stem cells is still in the basic research period. So we need the more fundings and the attentions, and we need the genius and scientists around the globe, the great minds, to put the joint effort to solve the problem, how to increase the rDNA. That will be the final step to increase the rejuvenation.
Okay, I have one last question. What about P53? Do you think manipulation of the level of P53 can give us the correlated benefits as if you increase the telomeres or the rDNA race?
Yes, thank you for the question, and this has been it's very being brought because people's first will be if we can. Well, yes, we can increase rDNA and telomere to decrease the P53 level, but can we just directly decrease the P53 level? Because as we know, P53, the protein, is the master control of the cell senescence. But actually, since telomere and rDNA, they are actually a driver or the countdown timer of the genetic program and the counters of cell division times. If we just try to rejuvenate aged cells by knocking out the P53 gene or continuously inhibiting the P53 protein, such as by antibodies, the gene expression profile will not change over time axis, and the cell division would go out of control, which will be the same property as cancer cells. Therefore, we can only knock down-regulated the P53 by lengthening the telomere and rDNA without causing uncontrolled cell division.
Okay, thank you. That's great.
Thank you.
Very good talk. Thank you.